Welcome everyone. I'm Dr. Shruti Analyx, Assistant Professor in the Department of Biomedical Engineering. So today we'll be looking at one of the topics in the ectoderm-derived organs, which comes under the Tissue Engineering and Regenerative Medicine course. So we're looking at a specific topic, which is the auditory system, the sensory hairs and their regeneration by need signals. So first coming to the auditory system. So this is the diagram of the auditory system. Shows basically three parts of the auditory system, which is the outer ear, middle ear and the inner ear. So the mechanism of hearing is as follows. The sound capture is done and basically the airborne sound waves are funneled in by the pinna. The pinna helps to collect the sound from various directions and they travel through the ear canal, which then strike the tympanic membrane. The mechanical amplification takes place in the next step. So the tympanic membrane vibrations can thereby vibrate the ossicles, which are the small tiny bones, namely the malleus, incus and stapes. So this will amplify the pressure at the oval window. So from the outer ear through the canal, tympanic membrane, and then it goes through the middle ear bones and the sound finally reaches the oval ear window. So here initial part is through air vibration, then it is through the bone vibration, and then finally from the oval window we will have liquid that is in the inner ear will be helping to transmit the vibrations over. So coming to the cochlear fluid motion, the oval window displacement creates pressure waves in the perilymph, which is the scala vestibuli, and this is the scala vestibuli, the top region, and from here sound waves further travel into the scala tympani. So this is the scala tympani, and this is the black region shown here is the basilar membrane. So the pressure waves in the perilymph is basically causing the bending of the basilar membrane. Next, coming to the sensory transduction. So the basilar membrane movement causes a shearing force on the hair cell, which is also called the stereocilia, and this is the organ of corti which is responsible for the hearing. In this stereocilia, there are mechanically gated ion channels which open based on the pressure variations. So these channels are mechanoelectrical transduction MET channels. So once these ion channels are open, so there is an influx of potassium and calcium, which leads to the depolarization, and thereby glutamate release is observed at the ribbon synapses, which are found between the cells and the neural junction, and this will activate the spiral ganglion neurons. So finally, the spiral ganglions will transmit the electrical signal via the auditory nerve CN8, and this will be transmitted through the brainstem, which is the cochlear nuclei, and then forward to the thalamus or the MGM, and from there it is transmitted to the auditory cortex in the temporal lobe. So this is the entire mechanism of hearing. So coming in particular to the specific uh, organ which is responsible for the hearing, which is the cochlea. So coming to a zoom version of the cochlea, we can see the scala vestibuli, which is the upper fluid filled chamber, and this is having the fluid perilymph, and this receives the sound vibration at the oval window. And there is also a lower fluid filled chamber, which is the scala tympani, and this basically ends at the round window to dissipate the extra pressure that is there in the ear. And there is also a cochlear duct, which is the middle chamber, and this is also called the endolymph, and this houses the organ of Corti. This is the organ of Corti. And basilar membrane, which is the flexible floor of the cochlear duct, and this vibrates selectively to the different sound frequencies. So there's also a tectorial membrane. This tectorial membrane is a gel-like shelf that touches the hair cells and shears against them to generate the neural signals. And then we have the organ of Corti. So this is the organ of Corti. So the organ of Corti basically consists of sensory epithelial cells, which are containing the hair cells, and this is helping to convert the mechanical vibration into electrical vibration. And then there is also the spiral ganglion, and these are a cluster of sensory neurons which help to transmit the auditory signals from the hair cells to the brain. And then finally we have the cochlear branch of N8, and these are the auditory nerve fibers, and they carry the sound information to the brainstem. Yeah. The scala vestibuli and the scala tympani contain perilymph. Both the fluids are similar to the extracellular fluid, and they have low potassium and high sodium. While the cochlear duct, so the cochlear duct is where all the mechanoelectrical transduction takes place. Here it contains the endolymph. The endolymph is a very unique fluid as it contains something to the contrary of the perilymph. So it contains high potassium and low sodium concentration, and this is essential for the hair cell mechanotransduction. So okay, this is a more zoomed in version of the auditory sensory cells. So as I told you, these are specialized hair cells which are responsible for detecting sound waves and helping with the balance of the person. These hair cells are located in the organ of Corti within the cochlea as an essential part of the auditory system. They convert sound vibrations into electrical signals for the brain to interpret the sensation of sound. So in this diagram, we can see the inner hair cell, and we can also see the outer hair cell. The inner hair cell is the primary sensory receptor which converts the sound vibration into electrical signals for the auditory nerve, and the outer hair cell, these are the motile cells that amplify the basilar membrane movement, and this basically helps in the helps to sharpen the frequency tuning. We also have different other cells. We'll just see through them. So the pillar cells, which forms the rigid wall of the tunnel of Corti, and this helps to provide the structural support. And we have the cells of Hensen, which are the supportive cells that secrete the endolymph and maintain the ion homeostasis. And we have the cells of Claudius. So these are the supporting cells. These are the supporting cells lateral to the Hensen's cells, and these are involved in the fluid regulation. And then we have the cells of Datus, which support the outer cells and hold their bases in position. And we have the cells of Bocher, which are located near the Claudius cells, and they maintain the endolymph composition. Then there are the reticular lamina cells. So the reticular lamina cells are the apical surface membrane structure which seals the organ of Corti and holds hair cell stereocilia in place. And there is also the tunnel of Corti, which is the fluid filled space between the inner and the outer pillar cells, and this allows the vibration freedom. So as we described, so we have two primary hair cells, which is the inner hair cell and the outer hair cell. So the inner hair cell is the primary auditory sensors, and they convert the sound induced fluid motion into electrical signals via stereocellular deflection, transmitting the data to the brain via spiral ganglion neurons. The outer hair cells are the dynamic amplifiers. So the outer hair cells rapidly contract and lengthen in response to the voltage changes powered by the motor protein prestin. And this mechanically amplifies the vibration by almost 1000 times, and thereby this enhances the sensitivity, which is critical for hearing soft sounds. So some of the key insights that can be got from this are that when there is a damage in the inner hair cell, this leads to the profound deafness, and so this causes irreversible signal loss. Whereas if there is a dysfunction in the outer hair cell, this will generally reduce the hearing sensitivity as observed in age-related hearing loss. So, yeah. So we talked about inner hair cells, which are the true sensory receptors, and these generally form the ribbon synapses with the spiral ganglion neurons, whereas the outer hair cells are the electromotor amplifiers, and they change length in response to the sound. So when we look at hair cell regeneration, in mammals, the hair cell regeneration does not take place spontaneously after birth. So, this is due to the loss of the proliferating capacity of the supporting cells. But if we are looking at non-mammals like birds and fish, so the supporting cells can transdifferentiate or proliferate in order to make up for the loss of the hair cells. So if there's any damage, the supporting cells will convert itself into the hair cells. So what are the supporting cells? Supporting cells serve as both structural scaffolds and latent progenitors in the cochlea, and this is capable of transdifferentiating into the hair cells when activated via specific molecular cues. They are present in the organ of Corti and are interspersed between and around the inner and outer hair cells, including the Data cells, the pillar cells, Hansen cells, and Claudia cells that we had seen in the image before. So coming to the regeneration by need signals. So we have different signaling pathway that can control the signaling pathways that occur in the stem cells. So need signals are nothing but the signals that are necessary to maintain the stem cell like characteristics. So one of the main signaling pathway is the North signaling. So for this signaling, the ligand or the molecule that needs to bind is the JAK2, and the JAK2 needs to bind on the inner hair cell. The receptor onto which this is binding is the Notch1 receptor, which is present on the supporting cells. And the binding of this JAK2 ligand onto the Notch1 receptor will determine the hair cell fate. While BMP, bone morphogenetic protein, and fibroblast growth factors specify the inner versus outer hair cell identity specifically. So, based on the specific concentrations, the supporting cells can differentiate into the inner or outer hair cells. So, the FGF8 in specific drives the inner hair cell differentiation, whereas the FGF20 can drive the outer hair cell differentiation. So, what are some of the key regulator molecules that are found in the auditory system? First is the ATO1 molecule. So, this is a master transcription factor which is essential for hair cell differentiation and regeneration. Next is the Wnt beta-catenin and the MYC activation, which can reawaken supporting cell proliferation and hair cell differentiation in the neonatal mammalian cochlea. Next, there are also some important growth factors, so some of which we discussed here. So, FGF, IGF-1, BDNF promote the survival, proliferation, and differentiation of nascent hair cells. Another regulator is the notch inhibition. So, pharmacological blockades such as gamma secretase inhibitors can overcome the lateral inhibition, which the differentiating hair cell sends notch signals to the neighboring supporting cells. This will basically prevent them from also becoming hair cells. So, when this notch inhibition is being overcome using gamma secretase inhibitors, they become free and they are able to convert into the hair cells. So, this will basically free the supporting cells to convert into the hair cells. So, next, coming to the therapeutic strategies that can be specifically targeted for hearing issues. So, combining the ATO1 overexpression with the notch suppression and specific growth factor delivery, such as BDNF and FGF, we may be able to restore the hearing ability in mammals. So, mammals, as I told you, they do not have the innate ability to regenerate a damaged auditory system, but we can try to overexpress these molecules like ATO1, or we can suppress the notch, and we can also include certain growth factors in the delivery vehicles, and thereby we may be able to restore the hearing ability in mammals too. So the challenges in this uh, type of a system are majorly with respect to the precise spatiotemporal control, which is very essential if we need to avoid the aberrant cell growth. As I told you, the entire organ of corti has very sensitive um, hair cells, and if we end up giving the signals at the wrong place at the wrong time, this could result in the aberrant cell growth and it could be counterproductive. So, harnessing these signals offers a promising route to regenerate hair cells, potentially reversing the sensorineural hearing loss. But we still have certain challenges that need to be addressed to completely be able to use these signals. So, the current research focuses on optimizing delivery methods such as viral vectors, small molecules, and ensuring functional integration of new hair cells. So, what are the scaffolds that we can use for the auditory hair cell regeneration? The scaffolds aim to restore hearing by providing structural and biochemical support for the cell growth. So I have already mentioned some of these scaffolds. So first we can use decellularized cochlear scaffold. So the cochlea will be removed from a deceased or a dead patient, and cells in the decellularized scaffold will be removed. So basically this will help to preserve the natural extracellular matrix that is found in the native cochlea. And then we can further add in the stem cells, and we can guide these stem cells for the integration and differentiation into the decellularized cochlear scaffolds. So this is one of the most promising techniques. So next, coming to use of hydrogels. So hydrogels in specific, something called the matrigel, can mimic the 3D ECM by giving specific growth factors and cues. Cells are able to orient themselves in a three-dimensional manner, and they can mimic the cochlear anatomy to some extent, and this can enhance the hair cell progenitor survival and differentiation. Then we can also use nanofibrous scaffold. So aligned fibers which can be produced using electrospinning can be used to direct the neurite outgrowth and also to improve the spiral ganglion neuron integration in the animal models. And we can also use biomaterial composites, as in we can combine conductive or bioactive materials. We can use specific molecules like RGD peptides, which can help to stimulate the regeneration process. So with this we are ending this session. Thank you.